Small changes in a cotton plant can sometimes be the first warning sign of a serious disease. This condition is commonly associated with the bacterium Xanthomonas citri subsp. Malvacearum, the causal agent of bacterial blight of cotton. The disease can affect leaves, stems, and cotton bolls. One of the earliest symptoms is the appearance of small, water-soaked or waxy spots surrounded by reddish-brown margins. On leaves, these lesions often have an angular appearance because their spread is restricted by the leaf veins. In some cases, the lesions extend along the major veins of the leaf. As the infection progresses, the affected areas become brown and eventually develop into dead tissue. Stem infections can be particularly destructive. Dark, rotting areas may develop around the vascular tissues, causing the bark to split or peel away. Plant tissue above the infected area may then die, resulting in premature discoloration and loss of affected branches. A white, waxy bacterial crust may sometimes develop over older leaf lesions or infected areas of the stem. Cotton bolls can also become infected. Infected bolls may develop boll rot, seed deterioration, and discoloration of the cotton fibers. Unlike the angular lesions commonly seen on leaves, early ball infections usually appear as round, water-soaked spots. As the disease develops, these lesions become sunken and turn dark, brown, or black. The disease is caused by Xanthomonas citri subsp, malvacearum, which can survive in infected crop residues and contaminated seed. Warm temperatures combined with high humidity and heavy rainfall create ideal conditions for bacterial blight to spread. The bacteria can enter plant tissues through natural openings, such as stomata, or through mechanical wounds. This is why outbreaks can become particularly severe after storms, especially when heavy rain or hail damages the plants. Because the bacteria can be carried on seed, using high-quality, disease-free planting material is an important first step in prevention. Proper seed processing, including approved linting and treatment methods, may help reduce the risk of seed-borne infection. Volunteer cotton plants that emerge from seeds left in the field after the previous crop can also act as an early source of infection, so they should be managed carefully. For biological management, products containing beneficial bacteria such as Pseudomonas fluorescens and Bacillus subtilis may help suppress the pathogen when properly registered and used according to local recommendations. Neem-based preparations containing Azadirachta indica may also be used as part of an integrated management program where appropriate. If disease pressure becomes high, additional treatments may be considered. Copper-based products can provide protective activity against bacterial pathogens when they are legally registered for cotton and applied according to the product label. Seed treatments should also follow locally approved protocols, as some chemical treatments mentioned in older recommendations may not be legal, practical, or appropriate in every region. For long-term protection, plant resistant or tolerant cotton varieties whenever suitable options are available. Regularly inspect fields for early symptoms and remove severely infected plants and other potential sources of infection according to local agricultural guidance. Maintain an open canopy where possible to improve air circulation and help foliage dry more quickly. Avoid overhead irrigation and do not work in the field while plants are wet, since moisture can encourage bacterial spread. Clean equipment before moving between fields, especially when working with areas where disease has been detected. If a field becomes heavily infected, harvesting promptly may help reduce additional losses. After harvest, remove or properly manage infected plant residues so they do not remain as a source of bacteria for the next crop. Crop rotation with non-host crops can also help reduce disease pressure over time. The key symptoms to remember are waxy, water-soaked spots with reddish-brown margins on leaves and stems, angular lesions restricted by leaf veins, dark stem rot, and round, sunken brown to black lesions on cotton bolls. Early detection, clean seed, resistant varieties, good field sanitation, and proper moisture management are important tools for reducing the impact of cotton bacterial blight. Follow the channel for more practical plant care knowledge.